Hello, my name is Luke Davison and I'm a Senior Research Assistant at Transpharmation Ireland. And the research that I'm going to be presenting for you today is entitled Evaluation of Human ex vivo and Rodent in vivo Models of Inflammation for CNS Anti-Inflammatory Drug Development. So just to give you a bit of background, a lipopolysaccharide, or LPS, is a component of gram-negative bacterial cell walls that acts as a ligand for TLR4, toll-like receptor 4, triggering downstream pro-inflammatory cytokine release. As a result, LPS is commonly used to study inflammation-associated disease through a host of biological models, including human peripheral blood mononuclear cells, or PBMCs, human whole blood cultures, as well as in vivo in rodents. So PBMCs have recently gained interest as a means to test novel anti-inflammatories for CNS disorders due to the mirroring of microglial CNS inflammation in these diseases in the periphery through PBMC activation and inflammatory protein expression. Whole blood then has the added benefit of allowing the identification of plasma protein binding of candidate therapeutics, which is a significant obstacle for drug CNS bioavailability when administered peripherally. The use of rodents for anti-inflammatory drug screening is well established and is the driving force in preclinical drug efficacy and toxicity testing for progression to clinical trials. LPS administered both peripherally and centrally can be used to induce peripheral and central inflammation, from which novel therapeutics may be tested for anti-inflammatory effects in the CNS. Here we show that LPS induces inflammatory cytokine production ex vivo in humans and in vivo in rodents, and that various anti-inflammatory therapeutics can attenuate this LPS-induced inflammation. So in terms of methods for PBMCs are obtained from healthy donors and pre-treated for 45 minutes with either the p38 MAP kinase inhibitor SP239063 or the TLR4 antagonist TAC242, followed by 24 hours of 100 nanogram per ml LPS stimulation. And inflammatory cytokines were then analysed via mesoscale diagnostics assay. For whole blood then, also obtained from healthy donors, and this whole blood was pre-treated for 1 hour with either a TAC242 positive control or a specific NLRP3 inflammasome inhibitor MCC950. This was followed by 4 hours of LPS treatment at 8 concentrations, plus or minus 5 millimolar ATP for the final 30 minutes of the 4-hour LPS stimulation. This was then cytokines were then analysed via mesoscale diagnostics assay. And then for the rat IP injections, they're pre-treated 2 hours before with dexamethasone at 1 milligram per kilogram PO, then 1 hour of 0.1 milligram per kilogram LPS IV, and plasma cytokines were then analysed via mesoscale discovery analysis. So for the first set of results, pre-treatment of human PBMCs with the p38 MAP kinase inhibitor SP239063 produced a dose-dependent reduction in LPS-induced cytokine expression. So it may be hard to see here, but the black bar indicates LPS, which is significantly increased for all cytokines measured compared to control. The red bar then indicates TAC242 pretreatment, which was able to significantly attenuate LPS-induced cytokine production. And then the blue bars indicate SP239 pretreatment, which produced a dose-dependent reduction in LPS-induced cytokine expression for all cytokines measured, barring IL-6. Then second set of results, pre-treatment of human whole blood with the specific NLRP3 inhibitor MCC950 significantly attenuated LPS and ATP-induced NLRP3 inflammasome activation. So for figure A, the blue bars there, we can see that increasing concentrations of LPS produce a dose-dependent increase in the expression of IL-1 beta, IL-10, TNF-alpha, and IL-6. However, when we add in ATP for the final half hour of the 4-hour LPS stimulation, that's the green bars, we see that there's a significant increase compared to LPS alone, only for IL-1 beta, and not for any of the other cytokines, which suggests that ATP is the triggering factor for the NLRP3 inflammasome activation. Then figure B, we can see that, this might be quite hard to see, but from the first grey bar on the left compared to the green bars, we can see that LPS plus ATP activation of the inflammasome is significantly attenuated by increasing concentrations of the specific NLRP3 inflammasome inhibitor MCC950, which was observed only for IL-1 beta and not for TNF alpha, suggesting a specificity for the NLRP3 inflammasome. Then pretreatment with dexamethasone significantly reduced LPS-induced cytokine production peripherally in rats. So pretreatment with dexamethasone, here we can see from the black bars, compared to control, there's a significant increase in TNF alpha, IL-1 beta, and IL-6 expression with LPS treatment that is significantly attenuated by pretreatment with dexamethasone. So to summarize, pretreatment of human PBMCs with the p38 MAP kinase inhibitor SP239063 produced a dose-dependent reduction in LPS-induced cytokine expression. Then pretreatment of human whole blood with the specific NLRP3 inhibitor MCC950 significantly attenuated LPS and ATP-induced NLRP3 inflammasome activation. And finally, pretreatment with dexamethasone significantly reduced LPS-induced cytokine production peripherally in rats. So to conclude, LPS is a potent pro-inflammatory stimulant that can induce robust inflammation both in human peripheral immune cell cultures and peripherally in rodents. Both ex vivo human immune cultures and rodents are useful models of inflammation that allow for novel inhibitors of a number of pro-inflammatory signaling pathways to be evaluated for anti-inflammatory efficacy in a semi-high throughput manner. So future studies will investigate the efficacy of anti-inflammatory therapeutics in disease patient immune cultures. So looking more clinically at multiple sclerosis, Alzheimer's disease, Parkinson's disease, and others. Thank you very much.